Hello, everyone, and welcome to the Precision Medicine virtual event. My name is David I. Smith. I'm a professor in the Department of Laboratory Medicine and Pathology at the Mayo Clinic. And I'll be talking today about the history of next generation sequencing technologies. The person who's going to follow me, Jonas Korlak, is the Chief Scientific Officer of Pacific Biosciences, and he's going to talk about a long-read sequencing technology. All of sequencing basically got started with the development of Sanger sequencing. There were chemical sequencing methods that were in use before this, but it was Sanger-based sequencing, which is illustrated here, that was really the birth of the ability to sequence more than very short stretches of DNA. With Sanger sequencing in the first generation, you do 4 separate reactions, each with a different dideoxynucleotide to terminate the chains at each of the A's, C's, G's, and T's. You ran the separate lanes, and then by reading up the lanes, you could ascertain the sequence. However, the real advance and what made this a possibility for capturing something as large as the 6 gigabase human genome, we needed the development of automated DNA sequencers. And this was sort of in 1999's the penultimate Sanger-based sequencer, the ABI 3700. And this machine was a 96 capillary electrophoresis machine that each time it ran was capable of generating 75 kilobases of DNA sequence. With the vision of Craig Ventnor, this is actually a room inside Celera Genomics, which was a private company that he was the head of. And this shows a room with 330 of these ABI sequencing machines. And this became the powerhouse whereby it was possible to sequence the DNA of none other than Craig Ventnor. Now, the cost to actually sequence the first genome was about $3 billion. Once you had all the machines and everything in place, it's about $200 million for reagent costs. The reason for this is twofold. The first is that you had to clone individual pieces into E. coli to make many copies. You actually needed about 10 to the 13th copies to do the Sanger sequencing. The second reason is that each individual reaction was 10 microliters, and when you have to do millions of these, the overall cost of these two together is expensive. So the question is, how can we bring genome sequencing to the masses? How can you dramatically decrease the cost of genome sequencing? And of course, it raises the question, how much for a genome before it becomes a part of standard clinical practice? For many years, while this was ramping up, there was the thought and the push that the $1,000 genome would be a great target. But even that was a phenomenal improvement in technology because you had to go from something that cost $3 billion to reduce it down to $1,000. But in order to even start on that journey, and actually I'm going to tell you by the end of today that we may have exceeded and been able to do a genome for even less than $1,000, but in order to make this work, you had to solve the 2 major problems of capillary electrophoresis sequencing, and that was the cloning of each individual fragment into a bacterial plasmid, and then the use of lots of reagents for sequencing. So the first thing is, how can one replace cloning? And it turns out there are 3 main strategies that have been utilized. 2 of them are PCR-based. The first is called bridge amplification, and I'll show you a slide of that. The second is called emulsion PCR. However, there is a third strategy that does not rely on PCR. PCR tends to put in artifacts during the amplification process. So this is a non-PCR-based strategy, which is linear amplification using a Phi-29 polymerase, where the pieces are cloned into an adenoviral vector, and it produces rolling circles to make copies. However, one of the problems with each of these is they produce different amounts of amplified template, and you need a platform that can sequence those limited number of molecules. So clonal amplification by emulsion PCR is shown here. What happens here is that if you emulsify water in an oil mixture, you will produce very tiny water droplets that are immersed in the oil. If you do that process and sonicate, but in the presence of very tiny beads, and they can be of different sizes, the beads, at a certain concentration, you will generate water droplets that contain a tiny bead in them. Now, these beads are coated with oligonucleotides that are actually complementary to oligonucleotides that you place on the ends of all of the DNA fragments that you would like to sequence, shown on the top left. So when you sonicate all of these together at a very low concentration of DNA, these beads are small enough that you can have hundreds of millions of them in a tiny microcentrifuge tube. And even if only 1 in 100 of the beads has a piece of DNA, you can amplify that piece of DNA on that bead and millions of them in this tiny little tube. The beads that have amplified DNA can then be purified from the beads that do not by centrifuge gradient centrifugation, and you can purify the beads that have amplified DNA. The second technique, a technique that does not require this water mixture, is known as bridge amplification, and this is illustrated here. In this instance, instead of coating the surface of a bead with oligonucleotides that are complementary to oligonucleotides that you're going to put on the end of the DNA fragments, the oligos are actually on the surface of something that is known as a flow cell. The DNA fragments are then placed so they have complementary oligonucleotides at the end of all of those fragments, and then you denature them and you flow the DNA fragments, as shown in the top middle slide, across the surface of the slide. One end of the oligonucleotide will find its complement, and then you'll have a piece of DNA sticking up, but it's base paired down at one end. However, the other end has oligonucleotides that are also complementary to other oligonucleotides on the surface of the flow cell. So as shown on the top right, you can actually produce a tiny little bridge that also has a template and a primer. And this is what you need, and it's ideal for DNA polymerase to make a copy that's shown on the bottom left. If you denature that fragment, now you have two fragments that are complements of each other. If you repeat this process about 30 times, where there was originally a single DNA fragment, there will now be a cluster of about 30,000. And on the bottom right, you can see that there is a whole series of clusters on the surface of the slide. This has many advantages over the emulsion PCR, the simplicity of it, and the lack of needing to do the very messy oil, water, emulsions, and the purification that comes afterwards. The third technique is, again, this adenoviral technique, where the tiny DNA fragments are inserted into an adenoviral vector, and then a Phi-29 polymerase rolls around the circle to make copies. Now, interestingly, this one produces the least number of copies. You get about 300 copies of the DNA each individual DNA fragment. Those 300 copies compress down to something which is called a nanoball, or a DNB, and these nanoballs can then be deposited into a surface upon which DNA sequencing will be performed. Now, in addition to the techniques to amplify the DNA, and this is all for the first generation, next generation sequencing, the next thing was how to reduce reaction volumes, because the 10 microliter reactions that are done in capillary electrophoresis use considerable amounts of reagents. So there's different strategies. There are strategies where you can put clusters on a slide surface, and that was really illustrated with the bridge amplification that I showed you 2 slides back. The second strategy is to have small beads either on a slide surface or within wells, and these, of course, are the beads that have been done with emulsion PCR. And all of this comes together with something which we're now trying to call, instead of next generation sequencing, even though that is its name, massively parallel sequencing, or MPS, because that's what all of these are. These are technologies that do sequencing on a massive scale. And basically, for example, the very first generation that I'll show you in a second, where the sequencing was done in a picotiter plate, which contained 1.5 million wells. So the very first next generation sequencer was developed by a company, 454, and their first machine was called the GS20, for the Genome Sequencer 20, because it was capable of doing 20 megabases of DNA sequence each time you ran the machine, which was dramatic improvement over the 75 kilobases on the ABI 3700. This is how the machine works. Again, on the left side, you sort of see how the emulsion PCR amplifies the individual DNA fragments on a bead. In the bottom middle, the beads that are purified can then be deposited into the wells. And then on the right, this entire thing is set up in such a way that the beads themselves are now coated with the enzymes that are needed for pyrosequencing the nucleus and the Basically, what you then do is you flood the entire picotiter plate first with the A nucleotide, and you ask the question, is there an A incorporation in any of the wells, which will be seen as a flash of light because of the enzymes from pyrosequencing. You then follow it with the next nucleotide, one after the other. Now, on this right side of this slide, you see the first generation sequence that can be determined with this platform. And it shows both the strengths and the weaknesses of the platform because one of the strengths is it's a relatively long read, 191 base pairs. However, what you can see is the incorporations that occur. If there's a single base, a single A, that will be incorporated. But if there are two A's or two T's or whatever, both will be incorporated and you'll get a slightly larger signal. If there are, and you can see this on the top left, if there are 4 G's, you get a signal that's about 4 times the size. This is fine if you're discerning 1, 2, 3, or 4 homopolymers in a row. But if you're sequencing something for a clinical sample, sometimes homopolymers can be 10 or 15 long, and this machine cannot discern the difference between 10 and 11, 12 and 13, etc. So the strengths and the weakness was very long reads. Actually, the 191 base pair read I showed you on the previous slide was increased to over 400 base pairs, and actually by the time this machine was fully mature, it was capable of 1,000 base pairs of sequencing. And there were improvements in the machine, but the weaknesses remain. The emulsion PCR, the fact that the nucleotides themselves are not blocked, so if you get a homopolymer stretch, you can't determine the sequence. And the last one was, even though this machine did improve 25-fold in its lifetime, there was limited headroom for further increases beyond a 25 improvement. And remember, we're trying to improve DNA sequencing to reduce the cost of $3 billion to below $1,000. So increments of 25 are not good enough. We need things to improve 1,000-fold or even greater. The second major platform that was developed was the ILLUMINA genome analyzer, and this is the first generation machine from that. This machine used basically the bridge amplification that I showed you before with all of its advantages as compared to emulsion PCR. Then, once you have the various clusters on the slide, the second improvement, which is a very important improvement on this platform, is that each of the nucleotides has a different fluorescent fluor attached to it, and the fluor is attached in such a way that only a single nucleotide can be added at a time. So you flood all 4 of the labeled nucleotides across the flow cell surface, and you ask, what is the first nucleotide which has been incorporated? You take a picture, and that is shown on the top right. The next reaction is a chemical reaction that chemically cleaves the fluorescent group from each of the single bases that have been added, and now it is capable of adding a second base, and you repeat the process 1 base addition at a time. You take a picture of that, and that is shown in the second thing on the right-hand side. And since you know the X and Y coordinates of every single cluster, the reading of the sequence is the positioning of that cluster. So, for example, for the one that's on the far left, the actual sequence of the first 4 bases or 5 is G, C, T, G, A, the first 5 bases. So that is how the sequencing is performed. Now, this platform had dramatic advances. The first generation genome analyzer was capable of a gigabase of sequence. So it was actually better than the 454 at the best that it could possibly be. But what it generated this by making sequence from 48 million clusters, 48 million separate DNA molecules that were amplified, but the read lengths were quite short. They were only 31 base pairs. However, over the next 3 years, the platform increased its output by doing 2 things: figuring out how to get greater numbers of clusters on the slide surface and increasing the read length. And once the read length increased beyond 100 base pairs, you didn't have any of the same problems that you had with these very short reads that it was originally capable of. The second generation machine from Illumina was the HiSeq 2000. This is shown here. This made some dramatic improvements and had much greater output. But eventually, with this type of platform, and this is shown on the bottom right, you generated so many clusters on the flow cell surface, which is shown on the top left, that it becomes very difficult to discern one sequence cluster from another. And then the next advance is shown on the bottom left, which is to not just have a flat flow cell surface, but to have a pattern flow cell technology and Basically, what you do here is the DNA molecules that are actually amplified in the well itself. If you see the bottom middle picture, that shows the type of signal that you see with the same density as the one on the bottom right, but it's so much easier to discern what's going on in individual ones, and it has greater capability for increasing this by simply decreasing the diameter of each of the individual wells. The instrument performance that has happened on this platform has been dramatic. When this started 2007 on the GA, this machine was capable of a gigabase, and on the left-hand side, that's 200 and 400 gigabases of sequence output. You could barely see it. By the time the GA was retired, it had increased its output to 80 gigabases. The HiSeq, the next generation, started at 200 gigabases, was almost capable of 400, but with the improvements in the pattern flow cells, they were able to crack the terabase of sequence output. Today, the penultimate currently, Illumina sequencing machine is shown here. This is the NovaSeq 6000. This machine is capable of running, I'll go back a slide. This machine is capable of running 3 different sizes of flow cells. Their highest output flow cell is capable of generating 6 terabases of sequence output. So every time you run this machine, it produces 6 terabases of sequence output. This is an improvement from less than a gigabase to 6 terabases, is a 6,000-fold improvement on this single platform. Now, there are a couple of other competing platforms. One of them is a company that's called TORRENT, and TORRENT does a completely different type of sequencing. They do sequencing on a tiny little chip that looks almost identical to a computer chip, because that's indeed how it's made. What the TORRENT chip, however, is, instead of an electronic computer chip, instead shown on the bottom right, is basically millions of tiny little wells, but each well is a sensor. They're linked up and tied to basically a pH meter, and it's important because every single time a nucleotide is incorporated, a hydrogen ion is released. So basically, if you amplify DNA molecules on beads, very similar to the beads from the 454, but smaller, you can basically do the exact same thing that you did with 454 sequencing, but instead of using the enzymes, to see the emission of light, instead you're going to look for the change in pH. So then by putting this all together, basically you flow A, C, G, and T unblocked nucleotides across it, and if one is incorporated, and this is shown on the right, you're going to see a change in pH in that well. That will be an indication that that base has been incorporated. There are a couple of different versions of this. The very original one on the top left is the PGM, the Personal Genome Machine. The one in the middle is the second generation. They already have a third generation, and they are evolving. Now, there are weaknesses to this platform that are identical to its weaknesses on the 454 platform. They're almost the same, except for how they detect the bases themselves. And those weaknesses are the emulsion PCR, the homopolymers, the fact that as they try to play catch-up in terms of the output on the Illumina machine, they're not able to do that. Their output is a fraction of what can be obtained on the Illumina machine. And right now, Illumina really controls the sequencing space. Somewhere in the range of 90% of the sequencing that's done across the planet is done on that platform. Now, there are strengths to the TORRENT platform. One, it has a quick turnaround time, faster than the Illumina platform. There's a lower input amount of DNA necessary for selection protocols, and for this reason, a couple of years ago, the National Cancer Institute actually chose the Personal Genome Sequencing platform over the MiSeq, the Illumina sequencer, because they could get more signal with less amount of input DNA. There's also viable solutions to simplify emulsion PCR. It's called the Ion Chef, which is an automated machine that does those things, and it has a flexible output. Now, the total cost of NGS turns out to not just be a single thing of DNA sequencing, but is a series of things. The first is the library preparation. And of course, depending on the type of library you make, there are different prices. Actually, the simplest library preparation of all is whole genome sequencing. Some of the other ones are actually more complex and end up being a lot more expensive. Now, Sequencing is the second cost, and this also depends on what you're doing. If you're trying to sequence an entire genome, a human genome, you need to do about 100 gigabases of sequencing. However, if you're trying to sequence a small gene panel, you need a fraction of that. Now, the third major cost is analysis, and this is a huge bottleneck of this entire technology because there are a lot of things that are involved in the analysis depending on what you're trying to see, and there are still need for improvement in this area. And the very last one, it's probably the most significant, but yet is one people rarely even consider, is the cost for storing this data. So when you're saying how much does it cost, or what is the target, for example, in bringing whole genome sequencing down to a cost, you have to realize that all of these must be added together. It can't just be $1,000 for the sequencing and then $10,000 for everything else. Now, whole genome sequencing is an extremely powerful tool to examine changes across the genome. It detects point mutations, but it also detects copy number variations, translocations, and deletions. But there are problems with it. Even though the NovaSeq has truly cracked the cost for sequencing below $1,000, actually the cost for 100 gigabases of sequencing on a NovaSeq is only $375. But again, as I just mentioned before, that's just the sequencing cost. How much does it cost for analysis? Very important question is finding alterations that are actionable, because the reality is that less than 2% of the genome actually codes for protein, and only a fraction of the proteins that may have any alterations do we actually have something that we can treat based upon that. And the other issue is the overall turnaround time. So what will it take for whole genome sequencing to take off? First, this depends upon which patients are getting whole genome sequencing. Are we talking about cancer care and treatment, or are we talking about the entire populations? However, whichever one you are talking about, the total cost must come down. Can we bring library prep costs under $50? Can we bring sequencing to at or around $100? And what about analysis and interpretation? So I think that the real goal, it doesn't have to actually be achieved, but it is something that is a game changer in how you think about it, is what if a genome only costs $100, cost all in? That would be a phenomenal target that would be transformational. Now, there is a problem when we live in an Illumina sequencing world, when a single company has such a strong monopoly, because the lack of competition does not serve the scientific or clinical community. Reagent costs can remain high. There's no reason for them to reduce those costs. The customer service doesn't have to be as good as if they had competition and felt that they had to treat their customers better. But how to bring down the cost of whole genome sequencing? Further, there's a lack of motivation unless Illumina believes they have some competition, and I believe they might. There is a company known as BGI, the Beijing Genome Institute, and they have a bunch of million genome initiatives. The Beijing Genome Institute is now housed in Shenzhen, China. They are also the CNGB, the China National Gene Bank. So contained within the BGI Institute in Shenzhen is this very large depository of samples, and they have plans to do 1 million human genomes, millions of plant and animal genomes, and as many bacterial and viral genomes as they can. Their platform is based upon a technology developed by a company called Complete Genomics. Again, they use the nanoballs to amplify the DNA to 300 copies, and these 300 copies form these tiny little balls which can be deposited on the, you can see this on the bottom right, onto a pattern surface. You can have so many, you can have hundreds of millions of these nanoballs across the slide surface, and then these can be done with a combinatorial probe method for doing sequencing. The next slide actually shows their machine, which has just been produced and is now being made available in several places around the world. This is the T7. This is a machine which can generate 7 terabases of DNA sequence per run. Now, what's important about all of these things is that today BGI can do whole genome sequencing, and this is what they charge when you send samples to them. And this is library prep sequencing analysis of $600, but they're planning on the T7 to bring the cost to $200 by the end of 2019, and they're setting up plans to develop a bigger factory scale sequencer where they could bring the cost of whole genome sequencing down to just $100. This is a big thing. So competition is good. As BGI ramps towards providing $100 cost all-in genome, Illumina will have no choice but to compete. And we've seen a reduction in whole genome sequencing from $3 billion to $600 in just 19 years. So is it possible to get to the $100 genome? Is it possible to think even that we could get to a $10 genome? Now, how will the $100 genome be a game changer? For $100, whole genome sequencing will replace small gene panels and even whole exome sequencing, because those technologies actually cost more just to capture the DNA than $100. Genomes could be sequenced at or before birth, and then whole genome sequencing could be the clinical test for cancers. Now, if the full genome only costs $100, that's not the only use that you can do for DNA sequencing. But when the genome does cost that little, all the other uses for DNA sequencing, next generation sequencing, like sequencing small gene panels, transcriptomes, and anything else you want to do also costs less. So this drives the cost of all these technologies to lower prices. So what are the problems? Well, what does our genome sequence tell us? What good will whole genome sequencing be for a healthy population? And will there be health coverage discrimination? These are important concerns, but the reality is, and this is an estimate, and I'm fully expecting that I will underestimate, that it's been quoted that 50 million fully sequenced genomes will occur by 2022. So whether you think these problems are big problems or insurmountable problems, the reality is that we're going to have a large number of people sequenced. Now, everything that I've been discussing is short-read sequencers. The Illumina sequencer does sequences of pieces that are about 250 to 300 base pairs. The problem the reason that you cannot sequence longer is that because you amplify the templates, when you're incorporating a base, you don't always get 100% incorporation. And when you repeat this over 100 iterations, what happens is the templates go out of phase and the signal strength decreases, and you can no longer discern what is the next base added. And it's quite difficult to assemble a human genome that's 45% highly repetitive sequences with 200 to 300 base pair pieces. In reality, what you're doing is you're generating a map of the genome of individual pieces so you can line that up against a reference genome. You're not generating an integrated genome. So the other issue is that it's really impossible to discern complex genomic alterations, for example, those that occur in cancer. And one last thing that I don't even have on the slide is when you're sequencing transcripts and looking at transcript isoforms, you can't tell one isoform from another with short reads. However, long-read technologies could do this. And this is known as second-generation massively parallel sequencing. And the idea behind it is to develop technology to sequence single molecules. There are two currently viable platforms for doing this. The first is Pack Biosciences. Jonas Korlak, who's the Chief Scientific Officer, will follow my talk and talk about that. And the second is a company called Oxford Nanopore. They use two different technologies, but they do the same type of thing. They generate sequences of single unamplified molecules, and because of this, and they're single molecules, the read lengths that they're able to generate are very impressive, in excess of 100,000 base pairs. So I will stop there, and then Jonas will follow up to talk about the second generation of these types of sequencing. Thank you.